Hello viewers, welcome to my channel Biopower. Today we are going to discuss an experiment showing oxygen is evolved during photosynthesis. Now this is the aim of the experiment, that is to show oxygen is evolved during photosynthesis. Now notice the setup very carefully. You have to take a beaker and procure some twigs of hydrilla. Hydrilla is an aquatic plant and it has not got any stomata. It will use the carbon dioxide that is dissolved in the water. It is generally found in pond. So aquatic environment, the carbon dioxide that is dissolved, that is taken up by the leaves, and it uses the chlorophyll to produce oxygen and of course food for itself, that is glucose. Now you see this is a short stemmed funnel. This funnel is a short stemmed funnel. You have to invert the funnel on the hydrilla plant and take a test tube full of water and invert it on the stem, and always keep in mind that the water level in the beaker should be above the stem of the funnel, otherwise it is not going to work. Now this whole setup you have to put in sunlight. Even while you are drawing the diagram, if it is asked, then also you have to show that this sunlight is coming from anywhere. Like the setup should be in the sunlight for about an hour or more, strong sunlight. So what you will notice after an hour, that small bubbles are being produced steadily. Small bubbles are being produced steadily from the cut end of the hydrilla plant or twigs of hydrilla, and you will see that the water level has gone down. Next step is that we are going to take off this test tube from here and bring a burning splinter near the mouth of the test tube. What we will notice, that the splinter is glowing brightly, showing that this is the oxygen gas that has been evolved during the process of photosynthesis. So students, this is the aim of the experiment, that is showing oxygen is evolved during photosynthesis. It is a short one, but there are few things I would like to point out in this experiment. One is that always cut the twigs in an oblique fashion so that the exchange of gases can take place, and also these are the precautions. You have to cut the stem or the twigs of the hydrilla plant under the water so that no bubbles get inside the xylem vessels, and as I have told you, the level of the water in the beaker should be above the stem of the funnel. Another thing is that preferably please take pond water to make the experiment work better. And if you are using a land plant, it is not going to work, because in land plant the stomata is there, which obviously is not adapted to function underwater, so it will not be able to take the gas dissolved in water, that is carbon dioxide dissolved in water. And what you will see that the experiment is not working. And sometimes you may be asked that how can you speed up the rate of production of bubbles. So in that case you can add a pinch of sodium bicarbonate in the beaker water. So you will see that the the bubbles, obviously it is the oxygen, so the rate of photosynthesis increases, and you can speed up the experiment. So that was all for today. Hope you have been benefited by this short content. If you have liked it, then click share and subscribe. Thank you.